Okay, let's dive right into this explainer. Today, we're exploring a deeply fascinating, hidden biological process, a microscopic sacrifice that is literally happening inside you right now. It's a story of ultimate teamwork, where individual units willingly lay down their lives for the greater good of your whole system. We are talking about the phenomenon known as apoptosis, or programmed cell death. Now, what if I told you that simply staying alive requires billions of your cells to intentionally kill themselves every single day? I know, it sounds completely paradoxical, right? We usually associate cell death with injury or disease or things going wrong. But the truth is, human life, health, and development actually rely heavily on programmed, intentional cell death. Without it, well, you wouldn't be functioning right now.
So here's our roadmap for today's explainer. We'll start with the good kind of death, then meet the demolition crew, look at how the signal is sent, what happens when the balance breaks, and finally, the future of medicine. Let's shrink down to the molecular level. Section 1. The good kind of death. Apoptosis versus accidental cell death. We need to establish right out of the gate that not all cell death is created equal. Apoptosis is a highly regulated, genetically controlled process. Think of it like a polite, perfectly controlled building implosion. It actively requires energy, the cell shrinks down, the outer membrane stays perfectly intact, and there's almost zero inflammation. Necrosis, on the other hand, that is a messy, random explosion caused by sudden trauma. The cell swells up, bursts open early, and causes significant inflammation and damage to the whole neighborhood.
Section 2. Meet the demolition crew. How a cell safely dismantles itself.
So how exactly does a cell pull off this perfectly controlled implosion? It actually happens in 6 really elegant stages. Once the cell receives that death signal and the internal pathways activate, specialized enzymes get right to work. The DNA and the cellular components are systematically broken down. But the really brilliant part happens next. The cell doesn't just burst, it neatly packages itself into little fragments called apoptotic bodies. Finally, immune cells called macrophages act as a microscopic cleanup crew. They sweep in, quietly consume these packages, and leave absolutely no mess and no inflammation behind.
Now, the real stars of this whole demolition process are specialized protein-destroying enzymes known as Caspases. They are the actual demolition crew inside the cell, and there's a very clear division of labor here. You've got your initiator caspases, like caspase 8 and 9, which are basically the foreman. They get the signal and start the cascade. Then they hand the job over to the executioner caspases, like 3, 6, and 7. These are the ones that actually swing the sledgehammers, tearing down the DNA and the cell's structural skeleton.
Section 3. How the signal is sent, the triggers, and the guardians.
Okay, all of this controlled demolition is fascinating. But how does the cell actually know when it's time to pull the plug? Well, the answer lies in 2 main pathways. First, you have the intrinsic or mitochondrial pathway. This one reacts to internal stress. If the cell detects severe DNA damage, oxidative stress, or radiation from within, it sets off the alarm. The second is the extrinsic pathway. This kicks in when extracellular death signals, essentially direct orders from the outside, bind to specific death receptors right on the cell's surface, like the FAS or TNF receptors. Let's zoom in for a second and see how this builds into a deadly chain reaction, specifically inside that internal pathway. When internal cellular damage is detected, the outer membrane of the mitochondria, the powerhouse of the cell, actually becomes permeable. It springs a leak. This allows a molecule called cytochrome C to escape out into the cytoplasm. Once cytochrome C is loose, it forms a structure called the apoptosome, which activates caspase 9, and boom, the point of no return is officially crossed.
But releasing cytochrome C isn't a decision the cell takes lightly. Cellular survival is basically a constant invisible tug-of-war, and it's governed by a group called the BCL2 family of proteins. On one side, you have pro-apoptotic proteins trying to promote death, pulling hard against anti-apoptotic proteins on the other side that are desperately trying to keep the cell alive. Ultimately, the balance between these specific proteins is what dictates the cell's final fate.
Overlooking this entire high-stakes process is arguably the most critical decision-maker in your cells, a protein called p53. Its primary day job is to constantly monitor your DNA for dangerous mutations. If it spots some damage, it has the absolute authority to hit the brakes and pause the cell cycle for repairs. But if that damage is just beyond repair, p53 pulls the fire alarm and immediately triggers apoptosis.
Because of this massive responsibility, p53 has earned a pretty epic nickname, the guardian of the genome. It's a protein that will completely sacrifice a single defective cell just to save the entire organism.
But tragically, when the gene responsible for p53 mutates and this guardian actually loses its function, it becomes one of the most common driving factors behind many human cancers.
Section 4. When the balance breaks, the stakes of survival. Because apoptosis is so profoundly fundamental to our health, the macroscopic stakes are incredibly high when this delicate, microscopic molecular balance fails. Think about it. If there is too little apoptosis, cells essentially become immortal when they absolutely shouldn't be. This is a classic hallmark of cancer. Cancer cells find these sneaky ways to dodge the death signal, allowing them to grow uncontrollably and form tumors. Similarly, if your body fails to eliminate self-reactive immune cells, which are immune cells that mistakenly target your own tissues, it can lead to some really debilitating autoimmune diseases.
But the flip side is just as devastating. When there is too much apoptosis, you experience severe, irreversible tissue loss. This excessive cell death is a massive contributor to neurodegenerative diseases like Alzheimer's, Parkinson's, and Huntington's disease, where precious neurons progressively die off. It's also the exact mechanism that causes the massive wave of cell death following ischemic injuries, like what happens right after a stroke or a heart attack. Actually, to really remind us of the sheer wonder of this system when it works perfectly, just consider this. During embryonic development, your hands and feet start out essentially looking like little paddles. It is the perfectly timed, programmed death of the cells between your digits that actually sculpts them, separating out your fingers and toes. You literally owe your human shape to apoptosis.
Section 5. The future of medicine. Hacking cell death.
Now that we understand the incredibly high stakes of this process, it makes sense that scientists are no longer just observing it. Modern biotechnology is actively aiming to selectively flip that apoptosis switch on or off depending on the disease. For instance, did you know traditional chemotherapy and radiation actually work by forcefully inducing apoptosis in cancer cells? Meanwhile, gene therapy is looking at ways to fix broken signals altogether, like repairing a mutated p53 gene. And on the other end of the spectrum, in regenerative medicine, researchers want to inhibit apoptosis to keep precious stem cells alive for tissue engineering.
And the cutting edge of this research? It is moving incredibly fast. Today, researchers are utilizing AI-assisted drug discovery to pinpoint brand new molecules that can regulate this process. The ultimate goal here is precision oncology, meaning personalized therapies that can finally overcome a tumor's resistance to apoptosis. They're even looking at utilizing advanced gene editing to completely rewrite a cell's faulty death mechanisms from the ground up.
So, just to recap the crucial takeaways, these are the molecular heroes and villains keeping you healthy day in and day out. From the executioner Caspases to the protective BCL2, all the way to the guardian p53. Understanding these components is absolutely the key to understanding how our bodies maintain that delicate, razor-thin balance between life and death every single second.
We'll end on this thought today. If science and medicine successfully learn to perfectly hack this system, forcing the bad cells to die and stopping the good cells from degrading, it fundamentally changes our entire relationship with disease and aging. If we can truly control cellular self-destruction, how close are we to cellular immortality? It's a completely fascinating question, and one that makes the study of apoptosis absolutely vital for our future. Thanks for joining me on this explainer, and I'll catch you next time.